Dear learners, in my last lecture I talked about light, pigments and the electron transport chain or light reaction. Just to summarize what happens in light reactions, some of the important steps of light reactions are absorption of light by photosystem 2, the flow of electrons in the so-called electron transport chain, which delivers electron to photosystem 1. When photosystem 1 absorbs light energy, it transfers its electrons to a small iron sulfur protein called as ferredoxin, and ferredoxin, by transferring its electrons to NADP, reduces it to NADPH. So NADPH is one product of light reaction, that is the reducing power or cofactor. Along with NADPH, another product is adenosine triphosphate. How is adenosine triphosphate or ATP generated? During electron transport chain, when plastoquinone is oxidized at cytochrome B6F complex, it means it transfers its electrons to cytochrome B6F complex. At the same time, it transports protons or pumps in protons from stroma into the lumen. The lumen is the space of the thylakoids. So within the lumen now there are two sources of protons. One is the oxidation of water, and second the protons pumped from stroma into the lumen. There is a higher concentration of protons within the lumen, but they cannot cross the lumen by itself. They need something to transport them from their higher concentration towards the lower concentration. And this is accomplished by another mushroom-like complex called as ATP synthase or ATPase. So using the energy of this proton motive force, the protons are transported from lumen into the stroma, and using this energy, the phosphate group is added onto a DP or adenosine diphosphate, converting it to ATP or adenosine triphosphate. So in this way, the ATP and NADPH, which are the energy carriers as well as the reducing power, they are generated in light reactions. So this is the first set of reactions called as light reactions, which generates ATP and NADPH. Now in the next set of reactions, by using this ATP and NADPH generated in light reaction, we will discuss how we can reduce carbon dioxide to carbohydrates. The next set of reactions has different names. It was called as dark reaction or light independent reaction for wrong reasons. This is a misnomer or wrong name actually, because we see that the next set of reaction depends on the products of light reaction, ATP and NADPH. So it does not happen, or it does not take place without using ATP and NADPH. So it doesn't take place at night. And it also depends on light for the activity of enzymes. So a more appropriate name is stromal reactions, because light reaction takes place in thylakoids, whereas the next set of reactions or carbon reduction reactions take place in stroma, that is the fluid medium surrounding these thylakoids. why this takes place in stroma? Because the enzymes required for carbon dioxide reduction are found in stroma. This is also called as photosynthetic carbon reduction cycle or PCR cycle. Carbon dioxide assimilation pathway or carbon dioxide fixation. So there are different names which you will find in literature. To simplify, we will use the term carbon reactions. Because in this set of reactions, carbon dioxide is going to be reduced to carbohydrates. So while light reactions require light, it is now clear that dark reaction does not require dark. It depends on the products of light reaction and the activities of enzymes. Many of the enzymes are light dependent actually. So now we have two sets of reactions, light reactions and carbon reactions, which work together to synthesize 3 carbon sugars or triose phosphates. This triose phosphate has 3 phases. It can be converted to starch within the chloroplast, but this is a reversible reaction. At any point, it can be converted back to triose phosphate. So this is a reversible reaction. This is phase 1. Second phase is that it can be exported to cytosol, where it can be used for the biosynthesis of carbohydrates as well as a broad range of other compounds that are required for plant metabolism, growth and defense. For example, the main form of translocation within the plant, sucrose, is synthesized. This sucrose can be transported to roots where it is used for respiration or generation of ATP. So starch, sucrose, respiration, they all represent different sinks and the partition. Third phase is regeneration of a 5 carbon sugar RUBP. RUBP or ribulose 1,5-bisphosphate is the first carbon dioxide acceptor molecule. This reaction is photocatalytic. About 1/6 of this triose phosphate is exported to cytosol, whereas most of this is used to regenerate RUBP. Why regenerate this? So that the photosynthetic carbon reduction cycle can continue. So before this is exported to cytoplasm, the cell regenerates the first carbon dioxide acceptor molecule, that is RUBP. As we discussed before that although plants have been photosynthesizing for so many years, but the details of this process of photosynthesis were revealed in the last 60 to 80 years. And one of the incredible and impressive story is the discovery of photosynthetic carbon reduction cycle by Calvin and his colleagues. carbon reduction cycle which occurs in all eukaryotic organisms in the honor of Calvin is also called as Calvin cycle, or sometimes it is called as Calvin-Benson cycle or Calvin-Benson cycle. Benson and Basham who were co-workers of Melvin Calvin. Calvin and colleagues performed a straightforward but clever experiment on a single cell algae chlorella, and they used something that was very new at that time, that is radioactively labeled carbon. Labeled carbon, that is C14. So first they eliminated the culture of algal cells, and they allowed the establishment of a steady state rate of photosynthesis, and then they supplied this radioactively labeled carbon dioxide or carbon. And by tracing this radioactively labeled carbon in the compounds that develop as a result of dark reactions or carbon reactions, they found that the first stable organic product was a 3-carbon compound, or 3-carbon sugar, called as phosphoglycerate. So, in their approach, they illuminated the algal cells, allowed to establish a steady rate of photosynthesis. Then they fed radioactively labeled carbon, and at different points in time, they would isolate the cells and put them in boiling methanol solution. What was the advantage of putting algal cell in the boiling methanol solution? There were two advantages. So it denatured enzymes. So it stopped the reaction where it was, because it was a time series reaction, and they could isolate the compounds that were formed and analyze them by paper chromatography. So this was the advantage of using boiling methanol solution. When they reduced this time to as little as 2 seconds, they were able to isolate the first stable product, that was a 3-carbon sugar called as phosphoglycerate. This was a 3-carbon sugar, or 3-phosphoglycerate. So most of the radioactivity was found in this 3-carbon sugar. That is why this cycle is also called as C3 cycle, because the first stable organic product of the first reaction of photosynthesis is a 3-carbon compound, phosphoglycerate, and the plants which only have the C3 pathway are also called as C3 plants. The examples of C3 plants are wheat, rice, soya. So now they knew that they used carbon dioxide and they got the first stable product, phosphoglycerate. That is a 3 carbon product, and there is one carbon dioxide. But still they had to solve another puzzle. The number of carbon in this first acceptor molecule of carbon dioxide. They didn't know what would be the carbon number in this first acceptor molecule. So from simple chemistry, what do you guess? You would be thinking it is a 2 carbon compound, and Calvin and his colleagues had the same thinking that it would be a 2 carbon compound. And the next 10 years they kept on tracing this 2 carbon compound, which they never found because it doesn't exist. Later on they found that actually this is a 5 carbon compound, that is RUBP or ribulose 1,5-bisphosphate diphosphate, abbreviated as RUBP. And instead of one molecule of phosphoglycerate or 3 carbon compound, actually 2 molecules of 3 carbon sugar are formed in the first reaction of photosynthesis. So this was the discovery of this ribulose 1,5-bisphosphate was the key to this crucial puzzle. So this impressive story of Calvin's approach or Calvin cycle is impressive because it highlights the importance of systematic investigation as well as patience in scientific research. Now we are going to discuss Calvin cycle. The Calvin cycle has 3 steps. We can divide this into 3 steps: carboxylation, reduction, and regeneration. Assume that it is cycle. So this is carboxylation step, in which carbon dioxide is added to a 5 carbon sugar RUBP, and the first stable product or 3-phosphoglycerate is formed. In the next step, this 3-phosphoglycerate is reduced to another 3 carbon sugar or triose phosphate, and this step is called as reduction. And the third step is regeneration of RUBP or ribulose 1,5-bisphosphate. So before carbon dioxide is reduced, first it is fixed or carboxylated. It is added to the first acceptor molecule or 5 carbon sugar RUBP. So Calvin cycle has 3 steps: carboxylation, reduction, and regeneration. Now I am going to draw the complete cycle. This is the carboxylation step, where carbon dioxide is added to a 5-carbon sugar, that is ribulose 1,5-bisphosphate, and it is converted to a 3-carbon sugar, 3-phosphoglycerate. This 3-carbon sugar, 3-phosphoglycerate, is converted to 1,3-bisphosphoglycerate. So this is phosphorylated using ATP and is converted into ADP and inorganic phosphate group. So where does this ATP come from? From light reaction. And where do these ADP go? To light reaction, where it can be used to generate more ATP. So let's consider that 3 molecules of carbon dioxide are added to 3 molecules of RUBP and 6 molecules of phosphoglycerate are produced, which are phosphorylated to 6 molecules of 1,3-bisphosphoglycerate. In the next step, using 6 NADPH of light reaction, 1,3-bisphosphoglycerate is reduced to glyceraldehyde 3-phosphate, or what we call as G3P. G3P is also a 3-carbon sugar. In some book, you will see PGAL or phosphoglyceraldehyde. So from here to here, this is carboxylation reaction, and from 3PGA to G3P or triose phosphate sugar, glyceraldehyde 3-phosphate. This is reduction process. Now we have to discuss the third step, that is regeneration of RUBP. So using ATP and NADPH of light reaction, we have reduced 3PGA to triose phosphate G3P, that is glyceraldehyde 3-phosphate. So as I told you that 3 molecules of carbon dioxide are added to 3 molecules of RUBP, so we make 6 molecules of 3-carbon sugar 3PGA, which are reduced to 6 molecules of G3P. Four, 5 and 6. So 1/6 of this is exported to cytoplasm, where it can be used for the biosynthesis of carbohydrates, as well as a range of other compounds for metabolism, growth and plant defense. This is converted via dihydroxyacetone phosphate. DHAP is dihydroxyacetone phosphate, also a 3-carbon sugar. This DHAP, dihydroxyacetone phosphate. This G3P is also converted to dihydroxyacetone phosphate, which combines with another G3P to form fructose 1,6-bisphosphate or diphosphate. This fructose 1,6-bisphosphate is converted to fructose 6-phosphate by losing one phosphate group, then to glucose 6-phosphate, to glucose 1-phosphate, and to starch. So we discussed 3 fates of this triose phosphate at the start of lecture, that it can be exported to cytoplasm or it can be used to regenerate starch. But this is a reversible process. At any point it can convert back to other sugars. So this fructose 6-phosphate combined with one G3P. So fructose 6-phosphate has 6 carbon, G3P has 3 carbon. So instead of forming 9 carbon molecule, this forms one 4 carbon molecule, that is erythrose 4-phosphate, and one 5 carbon molecule, that is xylulose 5-phosphate. This erythrose 4-phosphate combines with another G3P that is converted to DHAP, and now this 4-carbon erythrose phosphate combined with 3-carbon G3P to form a 7-carbon compound called as acetylose 1,7-bisphosphate. This acetylose 1,7-bisphosphate loses one phosphate group and is converted to acetylose 7-phosphate. And this 7-carbon compound, acetylose 7-phosphate, combines with the last G3P, and instead of 7 carbon of this acetylose 7-phosphate and 3 carbon of this G3P together, this makes total of 10 carbon, but instead of making a 10 carbon compound, it makes 2 5-carbon compound. Ribose 5-phosphate and xylulose 5-phosphate. So we have 3 5-carbon molecules now: xylulose 5-phosphate here, and ribose 5-phosphate here. all of these three are converted to the 5 carbon sugar ribulose 5 phosphate. So here we have 3 5 carbon sugar and they are converted to ribulose 5 phosphate, another 5 carbon sugar. So 3 molecules of ribulose 5 phosphate are made here. This ribulose 5 phosphate is converted into RUPP by utilizing 3 ATP. Here we use 6 ATP and here we use 3 ATP, which are converted to 3 ADP and inorganic phosphate group. So, a pretty simple cycle. I'm going to explain you in an easy way. The easiest way to explain this is in terms of carbon numbers. So here we have 3 carbons of carbon dioxide and 50 carbons of RUPP, because RUPP is a 5 carbon sugar and 3 into 5 is equal to 15 carbon and 3 carbons of carbon dioxide this makes a total of 18 carbons. And as I told you that phosphoglycerate is a 3 carbon sugar and 6 molecules of this 3 phosphoglycerate are made. So it means 6 multiplied by 3 is equal to 18 carbon. So we take 18 carbon and make 6 molecules of 3 carbon sugar. That is also the total carbon number remaining the same, 18 carbon. And when we convert this 3-PGA to glyceraldehyde 3-phosphate via 1,3-bis phosphoglycerate, this G3P is also 3 carbon molecule. And we make 6 molecules of G3P, 1, 2, 3, 4, 5 and 6. So carbon number remaining the same. And from here on, 1/6 of this or 1 out of 6 is exported to cytoplasm, whereas rest of this is used to regenerate RUPP. So here we are taking 5, 1, 2, 3, 4, 5, 3 carbon sugars and converting them into 3 5 carbon sugar. RUPP is a 5 carbon sugar. So we are taking 5 3 carbon sugar and we are converting it into 3 5 carbon sugar, 15 carbon. So this is how you can understand this better in an easy way. Why cells switch carbon? Because this is an easy reaction to perform. The free energy change is small and no high energy of activations are required. So biosynthetically it is an easy reaction to do, switching carbons among sugars. And the regeneration of RUPP is autocatalytic. It means before one sugar is exported, 1/6 of carbon is exported to cytoplasm. Before this step, RUPP is regenerated. Next, I would like to talk about the enzymes that are involved here. I will not go into the detailed biochemistry of these enzymes because I am teaching you from physiological perspective and how this cycle is regulated. So I will just briefly describe. Here in this carboxylation step, the enzyme that is involved is Rubisco. Rubisco is ribulose 1,5 bisphosphate, that is same 5 carbon sugar, but it is ribulose 1,5 bisphosphate carboxylase oxygenase. It means it can add or it can react with both carbon dioxide as well as with oxygen. So we have worked out how it reacts with carbon dioxide in C3 cycle or Calvin cycle. In next lecture I will discuss how it interacts with oxygen and what are the consequences of Rubisco adding or interacting with oxygen. Here in this step the enzyme that is involved is called kinase, that is involved with energy reaction. So the enzyme involved here is phosphoglycerate kinase. Here the enzyme that is involved is called dehydrogenase, G3P dehydrogenase, that is involved with redox reaction or oxidation reduction reactions. Here the enzyme aldolase is involved for reversible condensation of two sugars, and here transketolase enzyme is involved. I indicate here it has TK transketolase, and again here when erythrophosphate phosphate combines with DHAP, again aldolase is involved at this step. And here fructose 1,6 bisphosphatase enzyme is involved, which separates off one phosphate group, bisphosphatase. Here also bisphosphatase S2BP bisphosphatase enzyme is involved. Pseudoheptulose 1,7 bisphosphatase enzyme. Here transketolase enzyme is involved again. And for conversion of cellulose 5-phosphate to ribulose 5-phosphate, epimidase enzyme is involved, that is ribulose 5-phosphate epimidase enzyme, and for conversion of ribose 5-phosphate to ribulose 5-phosphate isomerase enzyme is involved. So as I told you that I'm not going to discuss the detailed biochemistry of these enzymes. I would like to share how this Calvin cycle is regulated. So from a physiology point of view, that is more important to discuss that how it is regulated. So here we see that for 3 turns of a cycle or for fixing 3 carbon dioxide molecule, we need a total of 9 ATPs, 6 ATPs here and 3 here, and 6 NADPs. So per molecule of carbon dioxide fixed, we need 3 ATP and 2 NADPH. Regulation of C3 or Calvin cycle. Calvin cycle is regulated by light, which causes changes in pH and by the redox reactions. During light reaction, suppose this is stromal side and this is lumen side. We discussed that this photosystem 2, and here we have plastoquinones, and here we have another complex, cytochrome B6 F complex. So during electron transport chain, plastoquinones also pump in from stroma into the lumen. And here, due to the activity of oxygen involving complex, water molecules split into oxygen electrons and protons. So now there are two sources of protons, and there is a proton-motive force is developed which causes higher concentration of protons on the lumen side as compared to stromal side. It means that the pH of lumen is lower. So now the pH of lumen becomes 5. Whereas pH of stroma, when protons are pumped from stroma into the lumen, it is equal to 8. So pH 8 means that light reaction is being performed actively. So Rubisco enzyme is acting, and pH 7 means the Rubisco is inactive. So neutral pH means the enzymes are inactive, whereas pH 8 means that the enzymes are active. So this is one way Calvin cycle is regulated by light or light reaction. There is another way. When photosystem 1 absorbs light energy, it can transfer its electrons to ferridoxin, that is in oxidized state, and it can reduce it. So now it is reduced. To remember oxidation and reduction reaction, oxidation is loss of electrons or protons, reduction is gain of electrons or protons. So remember these two words, oil and rain. So ferridoxin, photosystem on absorbing light transfers its electrons to ferridoxin, that is reduced to ferridoxin reduced form. Ferridoxin is a small iron sulfur protein. This ferridoxin can transfer now its electrons to NADP, as we discussed in the light reaction, and make NADPH. But there is another way. This ferridoxin can transfer its electrons. There is another small iron sulfur protein called as thyroidoxin. So here we have oxidized form of thyroidoxin, and ferridoxin can transfer its electrons to thyroidoxin, and on transfer of its electron, this thyroidoxin is reduced. So here we have an inactive enzyme which is in oxidized form, and this reduced form of thyroidoxin can transfer its electrons to open its bonds and to convert it into reduced form. And in its reduced form, this enzyme is activated. So that is how the for system 1 reduces ferredoxin, ferredoxin reduces thyroidoxin, and thyroidoxin can reduce an inactive enzyme by opening this disulfide bonds and to converting it to active form. So during night time, when light is not available, these active or reduced form of enzymes are converted back to oxidized form of enzymes or inactivated. They become inactivated. So this is another way how this Calvin cycle is regulated. So Rubisco and many of the other enzymes are regulated in this way by changes in pH and by redox reactions which are driven in principle by light. Now I would like to talk about the bioenergetics of Calvin cycle. We have discussed that per molecule of carbon dioxide fixed, we used 3 ATPs and 2 NADPH. Each NADPH stores 2 electrons, so this becomes to an input of 4 electrons. And this input is equal to, if we talk about energy efficiency, this is equal to 529 kilojoule per mole of carbon dioxide, while the output is 469 kilojoule per mole of carbon dioxide. So if one hexose sugar is oxidized, it will release this much of energy. So this becomes energy efficiency of approximately 88% or 90%. And if we include the input of ATP, that is equal to 282 kilojoule per mole, we add this to here. So now this becomes, energy efficiency of about 58%. But the underlying assumption is that all of the carbon dioxide remains inside the leaf. Under field conditions, however, this does not hold true, and actual photosynthetic efficiency is much lower. So to summarize today's lecture, light reaction requires light, but dark reaction does not require dark. Many of the enzymes of dark reaction are regulated by light. So dark reaction is a misnomer, or light independent reaction is a misnomer. So a better term that we can use is carbon reactions. Light reaction and carbon reactions are two sets of reactions. In light reactions, ATP and NADPH are generated, and by using those in carbon reactions, carbon dioxide is reduced. Before carbon dioxide is reduced, it must be fixed or carboxylated. So there are three steps of C3 cycle or Calvin cycle, the discoverer of photosynthetic carbon reduction cycle. That is carboxylation, reduction, and regeneration. And we have discussed in this lecture different steps how carbon dioxide is carboxylated or reduced, or how it is able to regenerate RUBP. And we have discussed that the energy efficiency is about 88%, and the regulation of this Calvin cycle is driven by light through changes in pH or through redox reaction.